Hello everyone, and welcome to my review of glycolysis. This review is not going to go over the mechanisms for glycolysis because there are some great videos on Learning Suite already that you can go through to learn about the mechanisms of glycolysis. Instead, what I'd like to focus on with this review is really bringing together glycolysis and gluconeogenesis, helping you understand how everything links together. And perhaps most importantly, one of the best ways to study glycolysis is to study it like a toddler. So we're going to be asking why for every step. Why does this happen? Why does that happen? Why do we care? And this will help you understand more about what exactly you need to know and why it's important. With this review, I posted it to YouTube specifically so that you can watch it at 2 times speed or whatever speed you'd like. Feel free to speed it up, won't be offended. But to start out, this is glycolysis. It looks big and complicated and scary, but we'll go through it step by step, hope you understand it. However, in order to get there, first we should figure out why we're talking about glycolysis. So, beautiful scene change there, I know, very impressive. So why do we do glycolysis? Well, this right here is the equation for cellular respiration. You've probably seen it before in other classes. C6H12O6 plus 6O2 goes to 6CO2 plus 6H2O. And that's really what this entire process of cellular respiration is for. Glycolysis is the first step in that process. But why do we take so many steps to get there? Well, if you look at these reaction coordinates, let's say we're doing cellular respiration the old-fashioned way, and that's by lighting a bunch of glucose on fire. If that's the case, the fire is going to provide the activation energy, and then we're going to go straight down, expend all that energy, come down here, and we'll end up releasing a ton of energy all at once. Why is that bad? Well, if we do that, that's fire. We all light on fire. We all explode. That is not preferable. We don't want to be on fire. So our body, instead, has developed a series of enzymatic reactions that looks something a little more like this. And I apologize for my left-handedness. I tried to write everything out a little bit beforehand. So instead of one giant reaction, we now have the same release of energy, but we're releasing that energy in bits over time. And with these small releases of energy, we can get intermediate molecules that'll come in and harness that energy. Molecules like ATP or NADH. So why specifically are we doing glycolysis to start this out? Well, there are a few main reasons. First, we can use enzymes to stabilize high energy intermediates. And then we can use those high energy intermediates to produce chemical energy in the form of ATP and NADH. Second, we could do a bunch of separate enzymes in order to learn and do gluconeogenesis, but our bodies are lazy. They're going to always choose the lowest energy route. So we do glycolysis because it's really easy to do gluconeogenesis and go back if we need glucose somewhere else in the body. We'll talk a little bit about that in this review as well. Final reason is to obtain pyruvate. Now let's take a quick look at pyruvate. Pyruvate is this middle molecule here. Important thing to notice about this is it's super oxidized. Look at all those oxygens there. And a highly oxidized molecule is a good reducing agent. Now, why do we care about reducing agents? Well, reducing agents, that's things like this here, NADH. Well, that's things like pyruvate, which will reduce things like this here, NADH. Reducing NADH, or NAD plus to NADH, results in the production of free electrons. These electrons are going to be taken into the electron transport chain later on, which we'll discuss a little bit later here soon in the semester. So that's why we like this highly oxidized product right here, because we can then take that and use it to reduce other things and take electrons into the electron transport chain. Now at this point, I'm going to go through all the different steps of glycolysis, and we'll just go through step by step, talk about what's going on, why we're doing it, and why the things have the names they have. So quick note, anytime you see something like this, a small phosphate, that's that. I know, beautiful transitions. So advanced. So this right here is a phosphate group. Important aspects of the phosphate group. Well, we've got a bunch of oxygen here, highly oxidized molecule. And we've got all these negative charges together. This phosphate, those negative charges are why ATP is such a high energy molecule. Because it's got a bunch of negative charges next to each other. That makes it highly unstable. And if you're unstable, you're ready to release energy. So ATP here is ready to release energy. So let's go into glycolysis. What's the first thing we're going to need to do? Well, we've got glucose in the cell. There's our glucose. It's beautiful. It got into the cell through insulin putting a transporter into the cell. Important thing about that transporter is it goes both ways. So glucose can come into the cell, but can just as easily go out of the cell. So what's the first thing we need to do? First thing we should do is make sure our glucose doesn't leave the cell. And what's the good way to keep something in the cell? Well, it's to put a charge on it. If you remember our phosphate group here, got two charges. So, in order to put a charge onto glucose, we're going to use hexokinase. So, hexo, that is a 6-carbon sugar. Kinase, anytime you see kinase, it's a transferase. All kinases are transferases, and it's going to involve ATP. Other important intermediate here, or other important cofactor, is this Mg2+. Now, Mg2+, what's the purpose of that? Well, Mg2+, it's going to stabilize these negative charges here and allow the phosphate group to jump off of our ATP and onto our glucose. Now, that phosphate group is on carbon 6. There's carbon 6, there's the phosphate, so we're going to call this glucose 6-phosphate. Easy enough to remember. So, take our glucose, we use hexokinase, and then we go to glucose 6-phosphate. Now, Dr. Graves wants us to know a lot of detail about glycolysis and about these reactions. If you look at his slides, there's information on each different enzyme. So, I would memorize all the information on those enzymes, everything on the slides. If it's on the slide, it's important. I know that's a lot, but glycolysis, we're going to come back to these enzymes. We're going to keep coming back to glycolysis. That's why he wants you to know it so well. And it'll also help you on the MCAT or the DAT or whatever other tests you may be taking for professional school, if that's the direction you're going. So for hexokinase specifically, it uses an induced fit. And an induced fit basically means we get glucose, we get ATP, we shove them together in the molecule, and then the enzyme actually changes its shape. So in the case of hexokinase, and most of the kinases we're talking about, when it changes its shape, it closes it off to the rest of the environment. That keeps any water from coming in and interacting with the ATP, which would cause problems. It could potentially hydrolyze ATP. Remember, ATP is a high energy molecule. It's pretty easy for ATP to decompose back to ADP. And that's why we use it to store energy. Compared to a lot of other molecules, it's pretty wimpy, but compared to a lot of biological molecules, it's super high energy. So we're going to induce that fit. ATP is going to come together in the active site, surrounded by positive charge on that active site to stabilize all that negative charge. Got that Mg2 plus stabilizing it. And we are going to get the phosphate coming in and replacing this hydroxyl group. So now we have a phosphate group there. All right, so we've got glucose 6-phosphate. What's next? Well, next we need to figure out a way, because we know we're going to split this molecule up down here into 2 3-carbon molecules. But this isn't exactly a symmetric molecule, and if we tried to cut it, we'd actually get a 2-carbon and a 4-carbon molecule, which we don't want, because then we'd have to use a lot more enzymes to figure all this out. And the body is lazy. We want to use as few enzymes as possible. So what we're going to do is convert this glucose into a fructose. The way you do that is phosphohexose isomerase. That again makes sense. We've got a phosphorylated hexose, 6-carbon sugar, and we're going to make the isomer. So isomerase is the class and the name of this enzyme. That isomer is going to be fructose 6-phosphate. Now notice both of these are in the alpha position. That's because our bodies only have the machinery and the enzymes to break up alpha bonds on sugars. So now we've got fructose 6-phosphate. Let me just double check if there's anything confusing that he wants you to know about phosphohexose isomerase. I think that's all really that you need to know on that, aside from what's on the slides. Still go in, look at his slides, and memorize that. But I don't think there's anything too complicated. Alright, now we've got a fructose 6-phosphate. So, we've got a fructose 6-phosphate. We're going to be cutting this in half soon. Well, if you cut this molecule in half, we're going to have half the molecule that doesn't have a phosphate group on it anymore. And remember, we need that phosphate group to keep the molecule in the cell. We need it to be charged, otherwise it'll just float out of the cell. So we better put a phosphate over on this side. The way we're going to do that is phosphofructokinase 1, or PFK1. Not to be confused with phosphofructokinase 2. That's going to be discussed when we're talking about regulation of these steps. So phosphofructose kinase 1, again, is going to use ATP. That's going to be led into ADP with a magnesium, and we're going to produce fructose 1,6-bisphosphate. So bis 2 phosphates. It's on carbon 1, carbon 6. Makes sense. The mechanism for this is a little different, but the enzyme works in the same way as hexokinase. And that's because it also works via an induced fit model. So now we have fructose 1,6-bisphosphate. Well, now we're going to want to cut this in half. How are we going to do that? It's going to be an aldose reaction. Now, you don't have to remember what an aldose reaction is, just memorize the mechanism for this. But it's an aldose reaction, so we call it aldolase. However, because these molecules are slightly different, we've got this right here, we get 2 separate molecules down here. So this one, let's think about why it would be named what it is. So again, beautiful visuals. This here is acetone. Acetone has 3 carbons, and it's got this ketone group here. Now if we look here, we've got those 3 carbons and a keto group, but we also have 2 oxygenated groups here. So you could call it dihydroxyacetone if that was just another hydroxy group. But in fact, that's a phosphate, so we just say dihydroxyacetone phosphate. And because that phosphate could be on either side and it'd be the same molecule, we don't have to label the number on the phosphate. Commonly, you'll see this labeled as DHAP, dihydroxyacetone phosphate. Over here, we've got glyceraldehyde, 3-phosphate. Now, if you look at glycerol, that is glycerol. So we've just got 3 OH groups coming off of 3 carbons. But here we have an aldehyde. So now it's glyceraldehyde. Oh wait, there's a phosphate too. We better label that. Which carbon is it on? Well, carbon 1 is going to be the one that has 2 oxygens bound to it, or 2 bonds to the oxygen. So carbon 1, 2, 3, glyceraldehyde, 3-phosphate. This is commonly called GAP. I've also seen it as G3P. I learned it as G3P. Dr. Graves usually will call it GAP. So, we've got 2 separate molecules here. Now we could take both these molecules, use separate enzymes, and go through to get pyruvate. But our bodies are lazy. So, let's be lazy and just use one single enzyme, triose phosphate isomerase, 3-carbon sugar with a phosphate, making the isomer. And let's convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Something interesting and of note with triose phosphate isomerase, it actually tends to favor this reaction. It tends to favor the production of dihydroxyacetone phosphate. But we're going down this pathway, so how the heck can that work? If we're making more of this, how do we do this? Well, it goes back to Le Chatelier's principle. So, because we're removing the glyceraldehyde 3-phosphate in the next step of glycolysis, we're going to favor this reaction, even though under standard conditions with both at equal concentrations here, it would favor the leftward reaction. So now we have glyceraldehyde 3-phosphate. And we've got this right here. If we put a phosphate there, we could get a really high energy molecule. Now, why would we want a high energy molecule? Well, so that we can harness the energy later. In addition, if we remove this hydrogen, this hydride ion, we could use that to reduce something else, to pass some electrons to something else. So that's why we're going to use glyceraldehyde 3-phosphate dehydrogenase. Sorry, the spelling kind of fell apart at the end there, but it's glyceraldehyde 3-phosphate dehydrogenase, I promise. So, or GAPDH is how Dr. Graves often labels it. So what are we going to do here? Well, it's a dehydrogenase, so we're going to get rid of that hydrogen right there. And we're going to do that using NAD+. Now, NAD+ is a hydride acceptor. So in this case, NAD+ is going to get reduced. Leo says GER, or oil rig. It's gaining the electrons because it gains both this hydrogen and the electrons in this bond here. So we're gaining those electrons. We get NADH. In addition, we're going to put an inorganic phosphate on the molecule as part of that process. Because we now have that inorganic phosphate there, we call this 1,3-bisphosphoglycerate. It's not a glyceraldehyde anymore because an aldehyde needs that hydrogen. So it's 1,3-bisphospho, again, 2 phosphates, bisphosphoglycerate. Now we can start producing some energy. So ATP is a really high energy molecule, right? That means it's pretty unstable. So in order to produce ATP, we're going to need an even higher energy molecule. We're going to need a super unstable phosphate. This phosphate, it does not like where it's at. It wants to leave. And so we're going to do that using phosphoglycerate kinase. Now wait a second, up here, hexokinase, and in the liver, glucokinase. That was taking the phosphate off of ATP. But you'll note this is a reversible reaction. And so we call this a kinase because it can go both directions. In fact, when you're doing gluconeogenesis, you are using ATP to produce an ADP and produce 1,3-bisphosphoglycerate. So phosphoglycerate kinase, it actually works, again, similarly to the other 2 kinases. It uses an induced fit molecule, acts as a transferase, but in this case, we are transferring the phosphate off of 1,3-bisphosphoglycerate and on to ATP. So now we've produced 1 ATP for glyceraldehyde. We have 2 glyceraldehydes. one that was this, one that was this. We converted them. We have 2 from our glucose. So we've produced 2 ATP, a net total of 0 ATP, because we used 2 up here and up here. So now we don't have 1,3-bisphosphoglycerate. We've taken the one off 1, so we'll just call it 3-phosphoglycerate. Notice this stays negatively charged. That's because of the pH in the body. At body pH, this will be negatively charged. It will lose its hydrogen just naturally. Well, now we've got this phosphate here, and we want to take that phosphate off, but remember, to take the phosphate off, we need a high energy molecule. This was super high energy. This, I mean, phosphate in general is high energy, but this is not a high enough energy molecule. We're going to need to produce something higher in energy. In order to produce that something higher in energy, we're going to first have to transfer the phosphate to the 2 position on this molecule. We'll see why in a second. So in this case, it's not an isomerase, it's a mutase. And it's phosphoglycerate mutase. Similar to an isomerase, works differently. Important with phosphoglycerate mutase, we get 2,3-BPG as the intermediate. Now, where do we remember 2,3-BPG from? 2,3-BPG, or bisphosphoglycerate, that worked in the hemoglobin to favor the T state. So why would that be? Well, if we're in muscles, we're doing a lot of glycolysis. It means we're expending a lot of energy. And so we're going to want more oxygen for later on in the process, in the electron transport chain, so we can produce more energy. In order to signal for that, 2,3-bisphosphoglycerate is an intermediate in this reaction. So when we're going from here to here, we produce this in the middle. And this can actually be released from the enzyme. So the enzyme doesn't have to go all the way through this. Sometimes 2,3-bisphosphoglycerate escapes. And when it escapes, it can then go interact with hemoglobin and favor the T state. So we release more oxygen and can produce more energy later on in the process of cellular respiration. But if that doesn't happen, which usually it doesn't happen, but happens often enough that it's significant, now we've moved our phosphate down here instead of up here. It's on carbon 1, 2, so it's 2-phosphoglycerate. Now, why did we do that? Well, we want to produce a high energy molecule. And if you remember from OChem, enols, wow, that one's even more beat up than the rest, but enols, look at that enol, what a beautiful enol. Enols are really high in energy. It's unstable, it's weird, cells don't like it, science doesn't like it, science likes it, but it's weird and it falls apart easily. So let's produce an enol. You know what we should call the enzyme that produces an enol? Let's call it enolase. So enolase is going to work, again, we're going to have Mg2 plus in there to stabilize things, and it's going to result in the removal of this oxygen, this hydrogen, and this hydrogen right here. He was hiding, but he was there all along. Ninja hydrogen. Ninja hydrogen is going to combine with those two. Basically, the enzyme is more complicated than that, but you can look at the mechanism. They're great videos. And H2O is going to leave the molecule or be utilized by the cell in some other way. And we're going to produce phosphoenol pyruvate. So this one is named that because it's really close to pyruvate, but we've got an enol and we've got this phosphate group, and the phosphate is replacing the OH in that enol. So now, oh man, that phosphate is so high energy, it does not like where it is. It wants to leave, and it wants to leave fast. It wants to leave really fast. So fast that this is an irreversible reaction right here. So we're going to use pyruvate kinase. And again, it's a kinase. but we're actually producing ATP. We'll be producing 2 ATP from 2 ADP. Why? Because we have 2 phosphoenolpyruvates. That's going to allow us to go to pyruvate, so we'll get 2 total pyruvates. And now pyruvate can be taken through the Krebs cycle, which we've discussed, and through the pyruvate dehydrogenase complex, which we've discussed. And we can take this high energy, highly oxidized molecule and use it to produce ATP for the cell to use. So pretty simple when you take it step by step. Now let's talk a little bit about the reversible reactions here. So reversible reactions are the ones that contain high energy substrates. That makes sense, right? We've got something really high energy to start it off. It really doesn't want to go back to that high energy unstable molecule. So in this case, ATP, high energy unstable molecule, it's going to make this reaction irreversible. Same thing here, ATP, high energy unstable molecule, going to make this reaction unstable and irreversible. And then here, it's actually the phosphoenolpyruvate that's our high energy unstable molecule. That phosphoenolpyruvate, it does not want to exist as that enol. It does not happy, and so it's going to do, that's what's going to lead this reaction to be irreversible. So those irreversible steps are going to be the locations where we have regulation of the cycle, which we'll talk a little bit more in a later video. It's also going to be the steps that we have to overcome for gluconeogenesis. As I mentioned before, one of the nice things about glycolysis is that we have all these reversible reactions. And because these are all reversible, it's pretty easy to go back and produce more glucose. More glucose that can then be exported out of the cell and utilized by other key organs in the organism. So to start out gluconeogenesis, really fancy now, we are going to use 2 enzymes. So we're replacing pyruvate kinase with 2 enzymes going the opposite direction. So we've got pyruvate, and we're going to use pyruvate carboxylase to produce oxaloacetate. That's what oxaloacetate looks like right there. So let's compare that to pyruvate. So it looks really similar to pyruvate, but we've added an extra carbon and 2 extra oxygens. And that comes in the form of CO2, as would be expected. How do we do that? Well, it involves this molecule called biotin and a carboxyphosphate. Let's learn about those. Whoa, transitions! So biotin, it looks something like this. There's our biotin. And you do not need to know the mechanism for this, but you should understand how it goes. There are actually a few different ideas. I would recommend learning all of them. You can learn more from the slide. I'm just going to go over it basically right now. So biotin, this thing right here, is going to bind to a phosphobicarbonate. And that just looks like a phosphate group attached to CO2. When it binds there, the phosphate group's going to leave, so it's just going to be stuck bound to CO2. It then brings the CO2 to another section in the active site where the pyruvate, that guy right there, is hanging out. CO2 then binds to the pyruvate, and that is going to form our oxaloacetate. So it's going to bind right there. And we're producing oxaloacetate because it's way too hard to take just pyruvate and take that back to phosphoenolpyruvate. This thing's way too high in energy. We need an intermediate that's a little bit higher in energy, a little bit closer in energy to phosphoenolpyruvate. That's going to require the cost of 1 ATP per pyruvate, or 2 ATP total per glucose molecule we're producing. Then we're going to use, oh, and I should point out, pyruvate carboxylase. So we're adding this carbon, this CO2, so carboxylase. If you see carboxylase, it means you're adding CO2 to something or taking CO2 off of something. In this case, we're adding it. Now we have oxaloacetate. We produce oxaloacetate. We can then take this oxaloacetate and convert it to phosphoenolpyruvate because we have a little bit more energy here thanks to adding these oxygens. And again, this is going to cost energy though. We're still going to need energy from GTP, which will be produced into GDP. This is basically the same thing as ATP. You can think of it as the cost of another 2 ATP, so this is costing us 4 ATP already. And we're going to call this carboxykinase. This enzyme here, PEP, carboxykinase, or phosphoenolpyruvate. PEP is phosphoenolpyruvate carboxykinase. So we're adding phosphate, taking off the carbon, the carboxy group here. So that's why it's carboxykinase. And we're producing PEP again. So we've got PEP. We can start going through this backwards. Just use that. We can just use that. We can use this here enzyme there. We can use this here enzyme. We can use triose phosphate to produce another dihydroxyacetone phosphate. Then those two can come together through aldolase. We've got fructose 1,6-bisphosphate, but oh no! This is irreversible. What are we going to do? Well, maybe irreversible, but we're smart. Our bodies are smart, rather. Or God. We can give God credit for this one. So, we're just going to use a different enzyme. We're going to take our fructose 1,6-bisphosphate, and we're going to say, hey, we don't like your phosphate here. We're going to take that phosphate off, because that's all that's happening right here. And so we're going to call it a phosphatase, because we're cutting off phosphate. And phosphatases, they are going to use water. They're going to hydrolyze that phosphate off. So we get inorganic phosphate coming off. We're going to call it fructose 1,6-bisphosphate ACE. You don't have to know anything about the mechanism for this enzyme. Basically just have to know this is what the enzyme's called and it uses water. That's pretty much it. Then that gets us to where we have our fructose 6-phosphate here. We can do that. That's fine. It's reversible, whatever. Got one more irreversible step. It's going to look a whole lot like the last one. So we've got glucose 6-phosphate. We're going to just call this one glucose 6-phosphatase, because sometimes scientists actually want to make sense with stuff they're doing. And so again, we're going to take water, take off that inorganic phosphate, and we're going to get glucose. And that is the basics of everything you need to know with glycolysis and with gluconeogenesis. I know it sounds like a lot, but if you draw these things out yourself, if you draw out the reverse yourself, and if you go through the slides to memorize the few things that he has on each of these different enzymes, you will do great on the test. This is a big portion of the test. It looks complicated, but an hour of study, y'all will have this down. I believe in you.